Hello there! This video is all about external validity of research methods, or being able to translate what you observe in a lab to the real world and to your population of interest. So it's really about going from samples to populations. So if internal validity is all about making true conclusions within the study, external validity is all about making sure the conclusions in the study are true in real life. So I will go over the general basis for external validity and then get into specific threats to external validity that can pop up in research studies. So again, external validity is really focused on how well will sample results be replicated in the population or replicated across different samples. And the level of confidence that results from one study would hold up in another study or in the population improve when you have a representative sample. So you can make a generalization from the sample to the population. So it's all about how well a sample represents individual differences in their population. It also depends on having realistic study features. So generalization from a research study scenario to a real world situation. And that depends on how similar the context is across the study to reality. So how well do the conditions of the study match the conditions in the real world? External validity also depends on the replication of findings with measures and manipulation. So making sure that the measures and manipulation of the independent variable function in the same way outside of the confines of the sample and the study. So let's take some time to focus on generalizing findings from a sample to a population. So wondering if the results observed in the sample would be replicated in another sample or even within the population at large. When it comes to concerns about having a non-representative sample, we're often worried about restriction of range. And restriction of range is when you really fail to capture the full spectrum or the full continuum or the full range of your variables of interest, and it often happens because you have a biased convenience sample. So a biased sample can happen if you rely on volunteers for your research, which is very common in human subjects research for ethical reasons. And volunteers may be more eager to please, more interested in science, more social, more outgoing, and have a whole host of other differences that are going to make them not necessarily reacting the same way or feeling the same way or responding in the same way as other people who didn't volunteer to participate in the research. Researchers from the University of British Columbia scoured all the available databases of comparative social and behavioral science studies, and they found that samples are weird. In fact, they found that people from, this is what WEIRD stands for, Western, educated, industrialized, rich, and democratic societies represent as much as 80% of study participants, but only 12% of the entire world's population. And unfortunately, WEIRD samples are not only unrepresentative of humans as a species, but on many measures, they're actually outliers. So you also know that a lot of research that's really geared towards helping or studying human behaviors and phenomena and treatments are conducted with animals for ethical reasons and for methodological reasons. There's quite a bit of internal validity when you do research on clones, right, on mice clones, because every single mouse is exactly equivalent and the same. But the external validity when you use cross-species generalization is pretty low sometimes. You really have to kind of study it in humans as well to be confident that the findings would generalize. So for example, there was research published that suggested, and if you look at the news article, it says teenage sex leads to bad moods later in life. So if you click that cross-species generalization link, it'll take you to that article. And they suggested that if teenagers have sex too early in life, they're going to be more depressed and anxious. But they studied this because you can't randomly assign teenagers to a sex condition or a no sex condition. They studied this with hamsters. So they took hamster clones and they randomly assigned 3 different conditions. 40-day-old hamsters who got to have some sex, which is like equivalent to a 16 to 20-year-old human. And then they looked at 80-day-old hamsters who got to have sex and a control group who didn't get to have any sex. the way that they looked at this was they really did find that those 40-day-old hamsters who got to have teenage sex were more depressed and anxious compared to the 80-day-old hamsters and the control group who didn't have any sex. But think about the way they measured depression. They measured depression by looking at how much energy those hamsters put into swimming when they were placed in water. And they measured anxiety as seeing how frantically they explored a maze. Not exactly sure how well those operationalizations of depression and anxiety translate to human samples. So you would need to maybe do a survey study with humans to see if the results are similar to what you replicated in the lab with the hamsters. So now let's focus on threats to external validity that involve the study features, and thinking about how we generalize results from a research study to a real-world situation. So how well will the results observed in this setting apply to other settings that my population may find themselves in? So think about how you would react or behave or feel in a lab setting versus a real-world setting. If you're in a research lab and you know there's a researcher recording your behaviors and monitoring what you're doing, you're probably going to be on your very best behavior. Versus if you're at home, you may not be on your best behavior. Hypothetical versus actual is all about really survey research. And so if you ask people to recall times where they were angry, or to recall their work performance, or to assess how good of a parent they are, they may not actually be that accurate, and they may overestimate how good they are. So it's nice to have observation, right? Because you have more of that internal validity. You can be confident that what you're observing is real, but you don't know if those observations would have external validity and would be how they would behave outside of the situation. So the novelty effect is basically just stating that participating in research can be a really novel, unique experience. And if you go to a lab that looks like the one you see here, maybe if they're trying to look at blood pressure or something, your blood pressure may be higher in that fancy lab without any stimulants ingested, just because you're in kind of a high stimulus environment that's really weird and unique and not what you're used to. Or another example could be if you have children and you're testing how effective learning is with VR versus standard classroom procedures, you may see decreased learning effectiveness and engagement over time as they become more habituated to the virtual reality. But at first, it's going to be very novel, and it may appear to have a really, really strong effect. So another feature of the study that may have an unintended influence on results are the characteristics of the experimenter or the researcher that's directly interacting with participants. So if different conditions have a different researcher involved, or even if participants within the same condition are interacting with a different researcher, this really could cloud your results. And it may not be the independent variable that's having an effect on your dependent variable, but it may be the type of interaction or the type of person that the sample interacted with. So for example, if you were looking at a study that was designed to test the effectiveness of different types of drugs on your ability to work out longer, right? They're testing these like workout enhancement or pre-workout drugs. And in the condition with the sugar pill, you have this sweet old man observing your behavior. And let's say that our population of interest are young men, right? And so our sample involves young men. Well, if the experimental condition with the supplement had the cute researcher from NCIS involved, and maybe they were into goth girls, maybe they would pump iron longer and they would work out harder to impress the girl, but not necessarily because they got the experimental drug. So another possible threat to external validity is the multiple treatment interference. So unlike the time-related concerns with internal validity that are about changes that happen between measurements, this is more concerned with the effect of the assessments or the treatments influencing the results. So for example, if you were doing a study where you were seeing if the Kaplan GRE review had a significant effect on GRE scores, and you did a pre-test, post-test quasi-experiment where you have students take a GRE, Then you have them do the review, and then you have them take the GRE the next day. Well, maybe the GRE scores are better on the post-test because the GRE review worked. It was effective. It helped their scores. But it could also be the fact that they already took the GRE once, right? Especially if it's the exact same version of the GRE. So they had some practice. They already knew what to expect, so they did better, right? It's hard to know what the effect is there in that study. Another one would be if you think about the movie genre, or looking at different clips from different types of movies and the effect on blood pressure. So if you are having exposure to various types of movies, the first movie you watch may have an impact on the effects of the other movies that you watch. So maybe if you're watching Saw first, it's going to raise your blood pressure and change the way that the other movies have an influence in your blood pressure. Or maybe if you watch Step Brothers first, then you're going to be in a lighthearted mood and you're not going to be as affected by the Saw movie. A way to figure out if multiple treatment interference is happening, especially within subjects' designs, is to counterbalance. So you see that there's two different conditions in that movie clip and blood pressure study. So you could have one condition that watched Saw first and another group that watched Step Brothers first, and then see if both conditions yield similar results, where you see higher blood pressure after watching the clip of Saw. Now let's take a look at how measurement issues and failure to replicate findings over time can have an impact on external validity. So if you remember, we have many different modalities of measurement. So you can have behavioral observation, you can do survey research, you can have physiological measures. And so measurement modality is all about how well does the way that you operationalize or measured your construct in the study translate to other measurements of that construct and to the real levels of that construct in the population at large. So it's all about how well you're tapping into the same construct with different types of measurements. So if you look here, right, you put on a cap to measure your brain waves, but how well does that really, those beta, alpha, delta, theta, and gamma waves translate to the things we think they do, right? So if you have more beta waves, does that really mean that you are more awake and that you're more alert, more conscious? Hope so, especially if you're using that as a measurement of being alert in a research study. So sensitization or reactivity is all about how the assessment itself, the measurement of the dependent variable, can make participants react differently to the treatment or the level of the independent variable that they received than they would in the real world or without the assessment. So a great way to make sure that sensitization is not having an impact on results is to use the Solomon 4-group design. And that's from the quasi-experimental and experimental research methods video. So having two conditions that receive the treatment, so you see A and C there, they receive the manipulation of the independent variable, with one that gets a pretest and one that does not get a pretest, allows you to control for the effects of the pretest on the response to the manipulation, to make sure that that initial assessment did not spur more awareness or sensitivity to the treatment that can influence the effectiveness of the treatment. So for example, if my hypothesis was that watching yoga with Cassandra videos for a week will improve your overall self-care routines, and I want to do this, so I'm going to study this by measuring self-care routines, the dependent variable, then have my sample do yoga with Cassandra for a week, and then measure self-care routines 2 weeks later. Well, maybe taking the first self-care routine made you more aware of what you should be doing to practice self-care, which made the sample more invested in yoga, which made the yoga have a stronger impact on self-care habits than just doing the yoga without taking the self-care inventory first. So that would be an example of them reacting to the measurement, and it has an effect on the way that the manipulation of the independent variable results in outcomes. Response latency is all about how well the effects that you observe in your research would hold up over time. So for instance, let's say that you have a family who's got a problem with screens, right? So on Friday night, they're all on their screens. They're not paying attention to each other. They're in their own little bubble, their own little world. And the next day, you have the parents read a book about getting your kids to play outside and why it matters, and you have them share that with their children. And then the following day, you see what types of activities they participate in. And your hypothesis would probably be that after being exposed to information about why it's so important to play outside, families would put their screens down and play outside. But you would need to do longitudinal research to make sure that the effects of that manipulation hold up over time, and so that by next Saturday they're not back on their phones. So a good way to do this is through longitudinal research and just making sure that effects hold up after a short period of time, and that they actually are lasting effects. Now let's take a look at one last threat to external validity, or how well the manipulation that you used in one sample would work in another sample or in the population. So here are some examples of concerns with manipulation, operationalization, generalization, or again, making sure that the intervention that you use that worked in the sample would work outside of the sample in other groups. So if you are trying to encourage physical activity with the reward of candy, right? You're saying if you participate in physical activity for 30 minutes, you'll be rewarded to see if people are more inclined to do physical activity. Zombies probably aren't going to be as motivated by candy as kids would be, right? So if your sample is kids, you would probably see a very strong effect. But if your sample was your population was zombies, the results wouldn't translate well because they want brains, they don't want candy. Another example is more realistic, and this comes from research that I would actually like to participate in and actually construct. So I kind of have a reputation for having a bit of a potty mouth in class, and so I want to do research to see if professors who use a little, like, smattering of potty language or foul language or cuss words, how would that effect that has on students' comfort in the class, feelings that the professor is a real person and not some sort of like automaton, their performance, their engagement, all sorts of outcomes. So if I had one condition at Nevada State University where the professor did not use any sort of colorful language, and then another condition where they did, and I looked at Nevada State University, I may not see the same results if I did that at Marshall University. I feel like, just through my experience, that the students at Nevada State seem to be a little more receptive to my colorful language, and I think the students at Marshall, it's a little bit more traditional, a little bit more conservative, were a little bit stunned by some of the words that I dropped in class. So that would be an example of the manipulation that you did in one setting may not have the same effect or work in the same exact way in a different setting. And I hope to do this research someday and actually collect the data and see if using colorful or even sometimes inappropriate language in class has positive effects on students.